welcome back to our podcast. Today we're joined by our special guest from I3L Fans. Hello, guys.
Hi.
Hello.
So today we're gonna talk about plants preservation, more in detail about plants preservation. What is preservation and how do it work?
The preservation is simply just we preserve things to keep access. The easiest one is the pressing and drying for the plants. The method is well known and has been established for many years. The how is it works? After the collection, the specimens are pressed flat between sheets of absorbent blotters and semi-absorbent paper. In this case, is newspaper. The plant should be carefully placed between the drying sheet because it will determine the apparent result. And the type of plant specimen will have different methods in placing them in the paper before drying. After drying, some specimen will turn into black, but generally it will turn into brownish or blackish color. If there's any contamination, it means the method is not performed well. The drying using microwave can be damage to the cells or cellular structure, therefore is not suitable for the long-term preservation. The alternative one is the silica gel or the seconds. Uh, it can be performed as the alternative. The shape and the chemical composition can be preserved in this method, but it can be into damage and the silica gel traces are difficult to be removed after drying.
Okay, so how about the unusual plants like algae or something?
For certain plants, the methods are not necessarily effective. Therefore, there are other methods to preserve such specimens. Fleshy and delicate plants are usually preserved in airtight liquid preservative. So it's usually composed of alcohol and water, but however, the colors will fade in this mixture. For smaller algae, usually formalin is used before storing the algae in microscopes like the view later. However, there are other plants who require special treatments. For example, cactus usually is killed before being dried or just put it in the alcohol solution. Like I said before, alcohol is actually used in a lot of these techniques. Bigger specimens need to be cut lengthwise before being pressed. And delicate flowers like orchids need to be placed carefully to prevent folds on the plant or just put it in the alcohol. And usually water plants are usually dried or preserved in formalin or alcohol. For larger plants like algae, it usually is mounted in shallow water. And lastly, in tropical conditions, usually the Schweinfurth method is used to prevent the specimen from rotting. This is actually just drying the plants with ethyl alcohol. These methods, however, usually doesn't work well with the fragile ones.
Okay, so how about the fragile ones?
So the process is called mounting, and what it does is it prevents the most fragile materials from fragmenting, and it prevents the specimen becoming set apart from the labels. The mounting method is conducted as the specimen should be mounted on a cardboard or an archival paper, and the fixing media such as the stitching with cotton thread, dental floss, nickel-plated copper wire for heavier specimens, or using an archival adhesive. So it is such as the metal cellulose adhesive. However, the disadvantage is that mounting can be hard for the specimen to be examined, so it is necessary to be careful during the arrangement of mounting process by using an easy to reverse mounting media, so the specimen should be trapped in the sheet, not in being glued over on the paper. I think that's all from me. Thank you.
Okay, so what Debbie and Linda and Jason said, it's just for a short amount of time, right? How about I want to put it in a museum? You need a long time, right? So how do it work?
So after we preserve the specimen, we must protect them for long-term preservation, right? So we must protect them for pest control, such as insect and fungi, which are mostly to make this specimen during the long-term preservation. The method that we use after we preserve the specimen is various, such as freezing, that is the most safest one, and then very simple, and then we can microwave it very fast, like 1 to 2 minutes. But as the said earlier, it can make some of it. Poisoning and insecticides use chemical substance, so it's a bit hazardous for human, but pretty effective against insect. And you can use fungal pest to control fungi with alcohol or diluted alcohol. For storage, you can store it in a cardboard or plastic box or in rain powder. It's up to you, but the pest control part is the most important part. So yeah, that's all for me.
Okay, nice. I learned a lot from you all. Okay, thank you. This is the end of our podcast. Thank you guys for watching this video. Bye bye.
Bye
Bye bye.
bye.